Remember, the whole point of a promoter is so that the polymerase, in this case RNA polymerase 2, can bind and then you can do transcription. So the question is, how does this happen? We're going to talk about the focused promoters, and we're going to talk about the 3 most critical elements in the focused promoter. That's on the DNA. We're also going to remember that the RNA polymerase doesn't bind on its own. There are a huge number of transcription factors, TFs, that help the polymerase bind to the DNA. And I'm not going to ask you to know what all these transcription factors are. We're not going to memorize the alphabet soup and how they bind. When you need to know that, you have to go look it up every single time until you're in a lab that really does this, and then you focus very specifically on it. But what we do want to know is, how does this whole complex find the DNA? And this promoter, these focus promoters, are only a little over 100 nucleotides long. So this is a relatively short piece of DNA, but it contains a few really important elements. The first one that essentially all focus promoters will contain is known as the TATA binding site, or the TATA box, which then binds a TATA binding protein, another transcription factor. So this TATA box is only 8 nucleotides long, and it includes a consensus TATA site. It's upstream of about 35 nucleotides upstream of the INR, which is the initiation or the initiator element. So this is where transcription will start, right very close to the initiator element. In fact, it usually encompasses the start site. So TATA box is essentially required for the transcription factors to be able to bind and the polymerase to be able to bind. It's upstream of the initiator element. And the initiator element also has a consensus sequence. So TATA has a consensus sequence that's TA, TA, and then 4 more nucleotides that have constraints. And again, the initiator sequence is about 2, 4, 6, or 7 nucleotides long, about 7 in humans, and that also comes with restrictions. So if you're scanning the gene and you see TA, TA, and then 4 more nucleotides subject to restrictions, and then about 30 odd bases later, you see this 7 base site subject to certain restrictions, then you can say, okay, that looks like somewhere where transcription factors can bind the TATA box, and that the polymerase II can actually start its transcription at this initiator region. There's one more element that seems to be common to all the focus promoters, and that is known as the BRE elements. BRE stands for TF2B, transcription factor 2B, recognition element. So it's called a BRE because the BRE recognizes transcription factor 2B recognition element. All right. So, and that tends to happen both right before and right after the TATA box. So again, these are short, somewhere around 8 nucleotide sequences that the TF2B protein can bind to. All right. The BREs, the TATAs, the INR, those are all cis elements because they're all on the DNA. The TF2Bs and the other transcription factors are all trans elements because they're coming from somewhere else. So that's the basis of a focused promoter. The basic elements are a TATA box, binds to the TATA binding protein, a BRE, which binds to the transcription factor 2B, and the initiator site, or the initiator element that allows for the polymerase to start transcription. There are multiple other elements that are often found, so DPE or downstream promoter elements, But right now what we want to focus on, and if you get nothing else, BRE, TATA, INR is where you need to start. Those are required to bind the transcription factors and the RNA polymerase II that's going to start transcription. All right, so we said that focused promoters are important to turn on genes that we only want on some of the time. And then I just spent 5 minutes explaining that they all have these certain elements like a TATA box, and not shown here, but you've got your BREs, and we said they're all kind of the same. Now you can imagine that certain TATA boxes, because there is variation, some of them bind more strongly than others, but this would still mean that some core promoters are stronger than others. And we said the whole point here is to be able to turn them on and off at will. And that's where the enhancers and silencers come in. So what's neat about enhancers and silencers are these pieces of DNA are a long way from the start site, the transcription start site. So they can be 10,000 or even 100,000 nucleotides upstream. Whereas your promoter, we're talking about somewhere on the order of about 100 nucleotides. These guys can be tens to hundreds of thousands of bases away. And basically what they are segments of DNA that are going to either help this whole initiation complex stabilize and bind to the DNA if you're an enhancer, or you're going to destabilize and knock it off if you're a silencer. So we've got enhancers and silencers that are regions of DNA that either increase transcription or decrease transcription. They're not required for transcription to occur. The core promoter is what allows transcription to occur, but they will modulate how much transcription occurs by up to several hundredfold, or maybe even more than that. So you will dramatically increase and decrease the amount of transcription product, that is the amount of mRNA you get based on your activators. So what we said, a couple of really important points here, enhancers can be far away. Interestingly, the core promoters, they have to be in particular order. So you have to have a TATA box. You can't have an AT-AT box. They have to be in order. You also have to have, so the enhancers, while we just said the TATA box has to be in order, the enhancers, you can actually pick the enhancer up and flip it over, and wow, that's an interesting arrow. But you can flip it over and it will still work. It's so far away that the DNA can bend enough that it doesn't matter which order it's in. So you can have a TATA box or an AT-AT box. You won't have TATAs and AT-AT for enhancers, but you can have an enhancer or a whatever that would be resynate, or however you say that backwards. You can put them in forwards or backwards. You can even actually have your enhancers within the coding region of the gene. So you can actually have enhancers and silencers within your coding region of your gene. So enhancers can be far away, very far upstream, very far downstream. They can be in either orientation. How do the enhancer sequences actually work? Well, one model, we're going to do 3 models, but one model, first model is what is known as recruitment. So here we've got an enhancer, and if this activator protein is present, then the enhancer allows the activator to bind to the RNA polymerase binding region, this whole protein complex, and allows it to start transcription. So maybe this activator protein responds to a heavy metal. So if there's some heavy metal, I don't know, mercury or something gets into your cell, then all of a sudden the mercury binds to the activator. Once the mercury binds to the activator, then the activator changes conformation and can bind to the enhancer and can bind to the whole complex, transcription complex. So when the activator is present, or when the activator protein is present with some cofactor, then transcription can occur. All right, so this idea of recruitment is there's actually something physically connecting the enhancer to the to the other transcription factors, and that allows for stabilization of the RNA polymerase initiation complex. All right, so this is probably the one we think about most often. There is some activator protein that is produced under particular conditions in the cell. So maybe that's heavy metal, maybe that is heat stress, maybe that is, there's a lot of CAMP. So the cell is starving, or maybe the cell has a high level of ATP, so the cell is in really good shape, and you would want different proteins involved in any of these particular cases. All right, so number one is recruitment, and obviously if you have a silencer, if you had your repressor protein, then somehow that would destabilize your complex. All right, so that's way number one that transcription factors can work with your enhancers to decrease or increase transcription. First way is physical recruitment. Second way that that enhancers and silencers can change the amount of transcription is through chromatin remodeling. So basically, in histone modification, so basically what happens is if you have a silencer or an enhancer region, given the appropriate environmental conditions, you'll actually go through and get your histone remodeling complexes to come in and change the nature of that local region of DNA to either allow transcription to start or to prevent transcription from starting, right? So you can either now allow your pre-initiation complex to come in, allow your RNA polymerase II to be recruited, or not, and that is dependent on, again, one of these enhancer or silencer regions, and then how it interacts with local conditions, whether that's heavy metal or heat stress proteins or whatever it is. When that region is activated or deactivated, it will change the histones. The third way that we imagine, or there's evidence that enhancers and silencers work, is actually really kind of fun. It's the nuclear relocation model. And often when we talk about the nucleus and talk about chromosomes, we say basically they're spaghetti within the nucleus. And it turns out that's kind of a lie. Each chromosome often has its particular location in the nucleus where most of that chromosome is. This is showing it is non-overlapping, and there's certainly overlap, like that orange and blue show overlap, but there's certainly overlap in these things. And there's another set of factors that are in here that some parts of the nucleus have a lot more RNA polymerase in them than others. So if here you have a region here that happens to have a ton of RNA polymerase, if your enhancer turns on and can pull part of this orange chromosome over here to where there's a higher level of RNA polymerase, you'll get more transcription occurring. So that's our third model of how enhancers and silencers work. They work by physically moving DNA around, because despite what Bio 1 says, the nucleus actually has a fair amount of organization in it.